Hello, my name is Jordan Avery, and I will be presenting on hypermethioninemia. So this is a genetic disorder in which there is an excess of a particular protein building block, an amino acid, called methionine in the blood. And so this disorder can occur when methionine is not metabolized correctly. So in our body, several enzymes break down methionine. However, with hypermethionemia, one of these enzymes does not work correctly, so this results in a buildup of that methionine in the body. So this condition is associated with other metabolic disorders as well, and those are considered primary, like hypermethionemia. Primary hypermethionemia. If the disorder is associated with another metabolic disorder, these disorders could be such as like tyrosinemia or galactosonemia. And so these can work in conjunction with hypermethionemia. Additionally, it can also result from excessive dietary intake of methionine as a result of consuming large amounts of a protein or methionine enriched formula. So the prevalence of this genetic disorder is a little bit hard to gauge. There is a limited number of cases that have been reported or identified, and the reason why it's difficult to gauge the amount of reports is because the symptoms are often not noticed or have no symptoms. So that makes it a lot more difficult to identify. However, those who are identified are typically identified through a newborn screening. So the major characteristics of this genetic disorder, though a lot of times those who do have this disorder do not exhibit symptoms, those would be delay in motor skills such as standing or walking, muscle weakness, liver problems, fatigue, and unusual facial features. And then another strange symptom is that their breath would smell like boiled cabbage, which is very strange, but that's just a little tidbit. So the responsible gene and chromosomal location. So hypermethionemia is caused by a genetic change in the AHCY, GNMT, or MAT1A gene. And so the reason for this being these genes are what tells the body how to break down methionine. And so if these genes are changed, the person will be unable to regulate the amount of methionine that is in their blood. Therefore, the person will have higher levels of the methionine in their blood, which will cause damage to the body and present the symptoms as I previously mentioned. And so the chromosomal location of this is on chromosome 20. So the mode of inheritance for this genetic disorder is autosomal recessive pattern. Typically inherited through that autosomal recessive pattern, that means that 2 copies of the gene in each cell are altered. And so typically parents of a person with an autosomal recessive disorder each carry 1 copy of the altered gene without showing any symptoms or presence of the disorder. Additionally, hypermethionemia can be inherited in an autosomal dominant pattern. And so this means that one copy of the altered gene in each cell is enough to cause the disorder. And in this case, usually one of the parents does have the disorder. So the functional or normal non-mutated protein. So methionine is an amino acid that is found in many proteins. However, one of the most interesting roles of methionine is that it can convert into a molecule called SAM, S-A-M. The molecule SAM can change your DNA by adding a methyl group. In regards to my chosen disorder, when methionine is properly broken down in the body, there is no buildup of methionine. So there would be no signs or symptoms of the genetic disorder. And so this is a pedigree representing the inheritance patterns of my genetic disorder. And so as you can see, those with the little red icon on them are those who have been afflicted with the disorder. And you can see that was represented by the lowercase p's. And so that will only occur if each parent has it but is not exhibiting symptoms, or one of them has it and one of them does not. But that does, just to touch back on that, the likelihood, as you can see, is very low with that autosomal recessive pattern. Luckily, just because a lot of these genetic disorders are very life-changing and can be very difficult for those who do possess them. So, mutations implicated in causing the disorder. So for this disorder, the mutations occur when there is an excess of methionine in the blood, and thus when the methionine is not broken down properly in the blood. So the protein encoded by this gene is an enzyme that catalyzes the conversion of S-adenosyl L-methionine, along with glycine, to S-adenosyl L-homocysteine and sarcosine. And so the protein is found within the cytoplasm, acting as a homotetramer, and so the defects in the gene are a cause of the GNMT deficiency. So in regards to genetic testing, so for hypermethioninemia, genetic testing is typically done in newborn screenings. And a newborn screening is when a baby's heel is pricked and a small amount of blood is then collected. And then a machine will run a test on that blood to determine how much methionine is in the blood. And if there are high levels of methionine in the blood, there's a possibility that the baby may have hypermethioninemia if they come back with high levels of that. However, that does not automatically mean that the baby does have it, rather that they need to be retested. And so these types of tests are usually done through a blood or urine test to determine if there is harmful levels of methionine in the blood. And so I find this quite interesting because I work at a pediatrician, so we have babies coming in every single day with newborn screenings. And I talked to one of the nurse practitioners today to inquire about those newborn screenings, and it was so interesting because though the amount of babies that come in or that leave the hospital, there's such a small percentage of those who do exhibit such like concerning genetic disorders, like less than 1%, those genetic disorders can be extremely harmful. And so that's why there's such a push on these newborn screenings. And so this is a diagram that I found that I really enjoyed looking at. This is from Baby's First Test, and so it kind of gives an overview of newborn screenings and what the newborn screening does. I mean, they're done. Every single baby in the United States is required by law to undergo newborn genetic screening prior to leaving the hospital, and this helps prevent serious or rare conditions from being underdiagnosed, like I had mentioned earlier, or undiagnosed entirely, which could be very lethal for children. And so the genetic testing, as mentioned here, is typically done 1 to 2 days after birth. And this is because this helps identify potentially life-threatening concerns that need to be addressed, proactively addressed in the first few days of life. And so this is my resource references page. I really hoped you enjoyed my project, and if there's any questions, please just send me an email.